Okay. We are reviewing chapter 17.
Oh, she's gonna be doing this.
Yeah.
Okay. Damn, you're not.
So we'll do a little game again this time. The games are just easier. There's less expectation there. Nobody has to know how many you get wrong. It's like, it's fine. So we'll do that. That's cool. Remember, like if we're doing this where I'm gonna go over my notes with you guys, essentially that this is gonna be silent time, if we will, on the Kahoot, but that doesn't mean you guys have to be silent. You can talk or look over your notes or whatever. It's not a quiz, right? So when we do the Kahoots, it's always like free reign. My other classes, I'll really like pass them around the note, like the sign-in sheet, and we'll be like, and I'm like, this is a Kahoot, guys. It's not a big deal. So you guys signed in, we'll get going with that. Chapter 18 should go by pretty quick. The hope is, and I know people will probably not show up until 11:30 in the lab, and that's fine. That's cool. But if we can get done early with chapter 18, then we'll just head straight up to the lab and get started. And then if people show up late, then they can just get started when they get started, man. It's fine. Yeah. The whole point is try to get like that lab going quick for my earlier classes, for my Monday morning, and then my Monday afternoon, and then my Tuesday morning, so that on my Tuesday evening, because tomorrow is Halloween, everybody wants to go home and do trick-or-treating with their kids. And Tuesday nights, lab doesn't usually get out until 8:30. So then I'm like, the sooner we can get done with chapter 18, the sooner we can get done with lab, the sooner we can get home to Halloween. So we can get everybody cranking quick on it. That'll be the goal.
No.
Yeah.
There's,
All right. I'll go ahead and start this. This is an easy one to join in if you haven't joined in already. Pick up get the right questions, pick up the treasure, don't grab the trash. Yeah, you see how quiet it gets. This is
no.
Almost $30.
Two. I'm gonna take one. Okay.
Awesome. Okay, hope that was a good little review for that chapter. We're going to go ahead and flow system resources. Great. Get into chapter 18. Let me get out of this really quick. Come on, thing. I'm not doing this for my own health. Okay. All right. Chapter 18 is dealing with diagnostics, and it's pretty much a lot of it is review of things that we have already learned. Honestly, like if you have looked at all at chapter 18, some of the stuff's going to be talking about PCR and some of the biochemical tests that we're learning about in lab fall within these categories as well. So a lot of this shouldn't be new stuff, hopefully. There's basically 3 categories of tests that we're going to be talking about for our diagnostics, and that's going to be phenotypic. So we already know that's going to be what these bacteria are going to be expressing, not necessarily their genes and what their genes say, but what is being expressed by them. So what enzymes are they actually using? What enzymes are causing fermentation and things like this? And then causing, usually, like we've already learned in lab, usually that's going to be associated with color changes in media and stuff like that. pH changes, whatever. Then we have immunological methods. We have already learned that has to do with antibody antigen interactions, always. No matter what kind of immunological test we're talking about, it's always going to have to do with antibody antigen interactions. And then genotypic tests. That's, again, looking at the genetics of an organism. That's going to be PCR and nucleic acid, other nucleic acid amplification techniques, which they call NAATs. If you guys are putting together your presentations, sometimes when they talk about diagnostic techniques, they say NAAT. That just means amplifying the DNA. That's all it means. So those are commonly used, as well as those hybridization techniques. Remember, if we think back to that one, we learned about like genetic engineering and stuff. Hybridization is dealing with making bits of DNA or RNA, little probes that are labeled usually with fluorescent labels, that if they stick onto sequences of DNA or not, tells you if that organism has that sequence or not. So that's what our probes are going to be working like. So it's the idea of our 3 main categories. And honestly, as far as like I feel like this chapter goes, that's like the gist of the whole chapter. But there's also been this move, of course, towards point of care diagnosis. If you guys, any of you who work in hospitals or even in vet clinics, they often rely on like point of care as far as like you take a little bit of blood, you put it on this little cartridge, and there's like usually a machine you can plug it into, and it gives you a result right there on that blood right there. That's point of care. So that the nurse, and you don't have to rely on the doctors, you don't have to rely on the lab, the nurse that is giving the care right there, who's treating the patient more accurately than any of those are able to do those tests right there with that patient and get the result immediately. Well, within maybe a couple minutes, but however long that machine takes to read it. We can even do things like A1Cs as point of care diagnostics now, looking to see if somebody's risk of diabetes is high. That was a common one that we would have tests for at Southwest. We had this like at the lab, we kept the cartridges and the nurses would just call for orders for like boxes of cartridges, like once a month or something. And they were good to go after that. So that was pretty common when we got was A1Cs. You can even do things like whole panels like CMPs and BMPs. They're not as reliable at point of care, but if you want to get a general picture of if this even is a concern to draw blood for this, point of care is great for that sort of stuff. So this is our little rundown table of what we're about to get into. Again, good for reviewing. First of all, you have to collect your samples. Again, these are slides I feel like are duh moments, okay? Like what kind of samples are you gonna be collecting? It might be blood, stool, you might get swabs from inside of the mouth or spit or sputum itself. I'm having somebody cough up phlegm into a cup versus just regular old spit versus swabs in the throat or swabs in the mouth on the cheek or whatever. Urine, either via catheter or clean catch mechanisms. You know, feces and all of these things, skin scrapings, biopsies. I think most people know what this stuff is. This guy down here, if you haven't seen, this is a typical swab that you might use to collect something that you're concerned about infection. So it's got a swab inside, like a Q-tip. That little red cap twists off and it breaks a seal. So you know that the seal's been broken and it's been used. If you're going to swab a patient, number one, first thing, if you're pulling out a swab, it should be sealed. If it's not, you're going to assume it's been used by somebody. So they break the seal, you pull the swab out, you swab the patient on whatever, and then put it back in here. There's usually a little area at the bottom down here that has media in it. It's what we call transport medium. It's not necessarily to grow it, but it's to keep whatever you swabbed happy until we can get it to DLO's lab or whatever to do growing of the specimen and all that. So we can actually swab the plate and do our quad streaks and things like that in the lab for people who are doing, you know, point of care specimen collecting, things like that. So you would swab, put it in there. This was a little like ampule that you'll like crack it to make sure it releases the media so it's nice and fresh. And then you should have already labeled it. Number one, when you're a nurse, probably number one thing you're going to learn is you label your tubes and your specimens usually at bedside while you're doing the thing and showing the patient the result, or you label them beforehand, depending on what setting you're in. They have their own SOPs, but you're never going to be leaving the patient and then labeling it. That's the absolute biggest no-no that you could ever commit. That's assuming you get busy. So many people get busy and haven't labeled their tubes, and then all of a sudden you don't know whose was whose, and you hope that you know. And we've gotten samples like that at Southwest before. That clearly people had mixed up their tubes before. So anyway, so we have forms that used to be used. This is not something that you really see a whole lot of in today's day and age, unless you're working in more rural or smaller clinics where they have paper forms that you'll be filling out to send off to the lab. 99.9% of the time, you're going to be doing this digitally through Epic or something like that. And it was used to be like Care 360, and I guess now most places have upgraded to Epic by now. So it's usually that interface that you'll be using. The same place that you're putting in your patient, you know, stats, their vitals, things like that, is where you'll be ordering your lab stuff. A lot of times as nurses, you may not be necessarily ordering the tests, but you'll be seeing that the doctor ordered the test, and you'll be printing the labels that are associated off of Epic, and then using those when you're labeling your tubes when you go collect the samples, whatever. It's the same concept as the paper, though. It's just done digitally. But essentially, there's all these tests that you could order, and knowing which tests you need to order or which samples you need to draw, because a lot of times it'll just say AFB, or it might just say AFB sputum. And you're like, cool, whatever that means. And so it's just a matter of knowing whatever terminology your hospital is using to do that stuff. If I were to say, like, I already have something like CMP or BMP, if you're working in a hospital and you don't know what that means, I guarantee you 100% within your first semester of doing rounds at a hospital, you'll know what CMP and BMP are. It's the number one most ordered blood test that you're ever going to have, aside from CBCs. Of course, everybody knows, most people know what CBC is. Complete, blood counts. So CBC is looking at your blood cells, CBC is looking at your white blood cells, CBC is looking at your hemoglobin, your hematocrit, and all of that stuff all at once, whereas BMPs and CMPs are metabolic panels. So complete metabolic panel and basic metabolic panels. So they're looking at your liver function, your kidney function, and things like that, enzymes that are associated with that. So anyways, those are abbreviations that you're going to see. And whenever you start getting, orders for that at hospitals, when you start doing rounds at hospitals, if you're not already working there, you will need to know what all that means and why is the doctor ordering these tests on your patient should be something that you're aware of as well. What kind of samples are being ordered and all of that stuff. It will, of course, become rote and routine for you working with that stuff. But man, it's a learning curve. So anyways, so we're talking about microbiology here. So our tests are going to be focusing mostly on infection sides of things. So you're going to collect your specimen that we're going to need to probably sometimes anyways, need to culture it. So we can culture it or we can do PCR immediately. And there are certain tests we can do right away, but occasionally we need to culture things. So you might want to choose if you think that your patient has an infection that's associated with gram-negative bacteria because maybe they have diarrhea. And a lot of times if you have bacterial infection in the stool, it's going to be gram-negative because those are the ones that survive the bowel, which we learned about with our McConkey auger and everything. So they're gram-negative. Maybe you want to use McConkey auger to enrich your specimen. You don't want any contaminants that might be gram-positive growing on there. So you might use McConkey auger to enrich that specimen. Man, if you get a stool specimen and we do, isolation methods and put it onto McConkey auger, and a grand majority of what's growing on there is, these creamy white colonies that are, this, and that probably means you're overgrown with that specimen. And that's probably our one of interest. So we can enrich for the thing that you're interested in based on symptoms, based on presentation, like where in the body this is occurring and all of that. Pick one of those selective or differential media to help us, pare it down immediately without having to start all the way from scratch from unknown with doing the quad streak and the gram stain and like all of that stuff. If you already are presuming it's probably this, based on what the patient's telling you in their symptoms, you can use the selective media to your advantage. And then the differential based on, okay, well, if it's, this organism, it's probably aerobic. We can do tests that deal with aerobic bacteria as opposed to anaerobic bacteria. So this is a dichotomous key. This is a very simple one. We have the one in lab that we're going to be using for identifying our unknowns that I need to update it because again, you guys have more bacteria than they did in the past, like 3 semesters because we couldn't get them to grow because we had issues with our water. This building is amazing. And so I need to update that. So you guys aren't going to get that probably until like the last experiment. I usually don't publish it until close to then anyways, because as soon as people figure out what their organism is, they think that they don't need to do any more of the experiments. And that's not true. FYI, everybody has to do all the experiments. There are people in here right now, maybe not in this class, because you guys didn't get the mycobacteria, but we have people in my Tuesdays and Thursday classes, or my online class, that they got the acid fast bacteria. If you had acid fast bacteria, you know that your bacteria is Mycobacterium something. And guess what? I'm not putting multiple Mycobacterium on that chart. There's only one species that I have that's Mycobacterium that you could possibly have. So if I gave them that chart, they already know what their gram-positive is immediately from that one test way back on experiment 4. So why would I give them that chart? They're just going to sit there and be like, I don't need to do any testing on my gram-positive now. But that's not the point of the lab, is it? The point of the lab is to see how the other tests work and what the results might be. So I don't need to deal with that. And I need you guys to be sure you're doing all your tests. So I usually don't give that out till the end, but this is what it will kind of look like. Ours is like not as vertical, ours is more horizontal. But if you had cockeye, if you did your simple stain and saw that you had cockeye, are your cockeye gram-positive or gram-negative? If your cockeye were gram-positive, let's say, so not gram-negative, and we're going to follow down this chart. Well, when we do the catalase test, was your catalase positive or negative? When you looked at it, did it have pairs or chains, or did it have irregular clusters or tetrads? Things like that. And then if it was one or the other, then you go down those pathways and then might do more tests following that. So was it an aerobe or was it a facultative anaerobe? So if you're facultative anaerobes, remember what those guys do? They ferment. So that's what we're learning in lab today, like our results today that we're going to look at from those tubes that we inoculated last time, whether they're yellow or made gas, means they fermented, right? So you do one of those tests, you find out that it can ferment. That's a facultative anaerobe. Only facultative anaerobes can ferment. So they If they don't have oxygen, they'll ferment. If they do, they'll go through aerobic respiration, right? So that's the preferred method of ATP manufacture. Wow, it all comes around, doesn't it? All the time. So one of my students, future students, came to my office last night, or I say last night, at 5 p.m. or whatever. And he was like, yeah, I'm coming into class. He's a paramedic. So he, has really random hours and wanted to be sure that he could fit it all in microbiology, and could he move between labs, because there's going to be like every other week he has random schedules, and like there's weeks where he's not going to be able to make Monday, Wednesday, and he's hybrid student, so that's Monday, Wednesday. I'm like, it's fine as long as you show up to your labs. I don't care if it's Mondays or Tuesdays or Thursdays or whatever it is, as long as you go to the lab that we're teaching. I don't care what day it is, and I don't make these accommodations for everybody. I make them for people who are already working in that field, working really hard, and can prove to me that their schedule is actually variant. If you're just, hey, some random thing happened in my life and I need to just always show up on Thursdays, that's a different thing, right? So he's coming to me ahead of time. I just want to be clear about that. But anyways, he's like, chemistry, it's just, we've been having issues with the biochemistry and the organic part. It's just hard. I can't wait to just get away from that. And I'm like, so about that. It's so yeah, and you guys have learned, like, it's not a ton. We don't do equations and we don't do math. That's the good part of it, right? We're not actually doing chemistry, but the theory is really important in this class. So I just reminded him of that. I'm like, hey man, if you can get the concepts behind these, that should be fine. But yeah, so that made me think of that. But fermentation, remember, we're talking about acid production, alcohol production, and gas production. If you show any of that, you can ferment. And if you can ferment, then you are facultative anaerobe. That's something you can use to identify your organism. So these things might be basic concepts we've learned about, but now we know not every organism does these things. So that means you can use it as an identifier. Okay, so that's what our dichotomous keys are based off of. So the first things that we are going to approach for this one are going to be our phenotypic tests. And our phenotypic test is going to be anything basically that is not antibody dependent or genetic dependent. So if it doesn't fall into those two, it's probably phenotypic. There are imaging tests we're going to talk about later that doesn't fall into any of these categories, but those are sort of like our random qualifiers. All right. So anyways, biochemical tests are what we're doing right now in the lab, looking at if you can ferment. If you can ferment, can you ferment these sugars? Can you break down starch? Can you break down urea? And all of those other things, whatever enzymes, it's usually enzymes, right? Let's be honest here, that you have that can break down crap. Like that's what biochemical testing is about. So these are physiological reactions in the bacteria. So the fact that you might have, I have brown hair. It's not actually naturally this dark, of course, but if I have brown hair and you had blonde hair, then that is a physiological expression. Something's going on where your proteins are making your hair blonde versus my hair brown. That's what we're talking about here for the bacteria. What enzymes are they making that allow them to do X, Y, or Z thing? And that's what we're testing for. Even the color of the colonies, or the fact that they're shiny versus matte or translucent, all of that stuff can be used as identifiers. So these are phenotypic things, even at the biochemical level. These are things that we will see expressed by these bacteria. Do they have a color change on the media? The color change itself is not the phenotypic part, right? It's the fact they make the enzyme or not. The enzyme being present leads to the color change on the media. But what really you're interested in is, does it break down lactose? Not that it turned pink on the plate, right? So that's what we're talking about here. So in real life labs, guys, in real life labs, they have these like cards or strips sometimes, just depends on what you're buying. So this is the API 50. So it has 50 tests on it. And each of these like sort of well things that they have here, like this one down here, number 40. This is like, for example, could be your urease test. So it's one of the things we should be inoculating today. It just depends on, my lab tech discovered that the filters that we need to filter that media, because we can't autoclave it because it's temperature sensitive. The filters we didn't have anymore. He like opened the box, it was just empty. And so we had to order more. So maybe we'll have urea broth today. Maybe we won't. If we don't have it today, we have to inoculate on another day. We're not just skipping it. So it's an important one. So yay, isn't this fun? But anyway, so let's say that's what that is. Urea. Can you break down urea? And it's a pretty easy chemical breakdown method for bacteria. If you break down urea, you give off as your products carbon dioxide and ammonia. So we've already talked about ammonia causing color change with phenol red, how it can make things turn pink, right? We're going to see that today, actually, in some of your results. That's what we use for urea too, urea broth. If you break down urea, you make ammonia, you turn the phenol red pink. If you can't break down urea, you don't have a color change at all. That's it. It's an easy one. But we can put tests like that one in all of our fermentations and everything in these little slot things on these cards and let the bacteria incubate overnight and then come back and look at the color changes on the cards, as opposed to each individual tube being inoculated and everything. So yay! And these are really expensive, which is why you guys don't get to do these. But so they're pretty expensive, cost prohibitively so for our purposes, but for labs that are needing to identify unknowns, this can be really useful. We used these a lot in my food testing lab. And that's what we're probably going to be doing with the cadaver stuff, because I just need more tests than what we have available in our labs to identify that one. But it's cool stuff. You get color changes. You just mark down on a piece of paper whether test 1 was positive or negative, or test 2 was positive or negative. And then you get numbers for those and add them together and plug it into a computer. And it tells you this is, Escherichia coli. And that's it. And that is really helpful, especially if you don't know what you're looking for, which would be the case if you really do have a truly unknown situation. You can't just plug it into PCR. PCR, you have to know what you're looking for. So anyways, we also have antimicrobial susceptibility testing. Kirby Bauer. So we already saw with our Kirby Bauer, if you guys actually looked at other people's plates with the little discs and the zone of inhibition, that some people had areas where they didn't have any susceptibility, they were completely resistant, completely overgrew on, like, let's say ampicillin, and it wasn't blocked at all. Other people had on their ampicillin, a zone of inhibition. So if I were to say, hey, this table over here had zone of inhibition and their gram negative was salmonella, and then this table over here, no zone of inhibition, completely resistant, and theirs was E. coli, you can start to see, those are both gram negatives, you can start to see how you could compare organisms based on how they react to certain antibiotics. This isn't always reliable because we can transmit the genes between certain bacteria with the sex pili, right? That conjugation aspect. But certain kinds of bacteria have reliable outcomes of what their antibiogram, the antibiotic susceptibility and resistance and whatever with Kirby Bauer. Some bacteria we can use that to identify with. Okay. So that's, again, presumptive identification. If you think this is strep instead of staph, maybe do Kirby Bauer. You're probably going to need it anyways to determine what antibiotic to give them. So you compare it to what you're expecting for one or the other. Another thing we can do, which is kind of similar, honestly, is phage typing. So this is where you take, instead of putting antibiotics on your bacteria, we're going to put bacteriophages on your bacteria. So you can create your lawn of your bacteria and create little segments in your dish and in your quadrants, and like put a dot of bacteriophage in each quadrant. This bacteriophage goes with salmonella. This bacteria only infects E. coli. This bacteriophage only infects this type of bacteria, and this bacteriophage, this one. And wherever you see killing of the bacteria, we see that as in the form of plaques. So it's going to be clearing areas in those lawns. So when those plaques develop, that means we're lysing open those bacteria with the bacteriophages, those viruses, and they only infect one species of bacteria. So it helps you identify that way. We don't use this, obviously, for every bacteria ever, but for some bacteria, it's easy to reach for the phage that we have and see if it infects it. If it does, it's E. coli. That sort of stuff. So phages only infect certain kind of bacteria. We can use that to identify. Well, if you're looking at the significance of our cultures compared to, right, so you're taking a sample from somebody's stool and you have some colonies on your plate, your TSA plate, your nutrient auger, that turn out to be E. coli. Is that abnormal? Probably not, right? But if the whole thing is like almost entirely E. coli, that might be a concern. So having E. coli present in certain areas of the body might not be a concern. Having E. coli present in areas where it shouldn't be would be a concern. having overgrowth of something where it should be a minor species is a concern, right? So we have to basically weigh things based on what you're expecting versus the norm. Because you might be, yeah, like it says, a few colonies of E. coli can be normal biota, but if you have just a ton in your sample, that can be a concern for infection. Whereas if you have any Mycobacterium tuberculosis, any tuberculosis bacteria in your sputum, you have an infection. Like that should not be there at all. So knowing that stuff is helpful too, because if you hadn't taken microbiology and you have someone come in that has a few little indicators of tuberculous bacteria in their sputum, you might not know that you shouldn't have any at all. So that's the idea. Being aware of that helps. The problem with a lot of these phenotypic methods is you may need culture time. Yes, we can do a gram stain on sputum directly. However, if that came back inconclusive, you still might need to do a culture of the sputum and then test it again. Culturing can take a minimum of 18 to 24 hours. Our cultures in our lab already go 48 hours. That's still more. So think of it that way. You guys just cultured your stuff 48 hours ago. Imagine if you were a patient waiting on your results for stuff, you know. E. coli, we can get that to grow within 7 or 9 hours. It's a very fast multiplier. But things like tuberculosis, if we need to culture that, are going to take like a week or so. So this becomes very prohibitive. Plus, the media you may need to enrich certain bacteria, you might not have on hand in your lab. You might have to order it, or you might even have to send certain samples off to the CDC or Los Alamos if we're talking about things like plague or Ebola or something that you don't see a lot of. So that can be very prohibitive if we're doing certain testing like that. So these are some examples that some of these we already went over for our phenotypic testing. Of course, direct examination, looking at where the infection is and how it's literally presenting with, like, you look at your patient, do they have a fever versus not having a fever? Or is there inflammation or not? That's your direct examination, and plus all the other ones that we've looked at. All right, on to the immunological stuff. This is also known as serology because we're looking at the immune, like antibodies, immune aspect of your serum. So anytime we're using serum for this, that's serology. This is going to be very specific. We already know. We've learned about antibody antigen interactions and how specific it can be. We're not looking for, is there bacteria? We're looking for, is there the bacteria that causes the plague specifically? So very specific immunological testing. In serology, we have our ability, now this should start to sound familiar, guys. Unknown antibodies can be detected using known antigens, or unknown antigens can be tested using known antibodies, right? So if you don't know if the person has the sickness, the illness, the bacteria, you better have an antibody to test it if you're doing immunological testing. If you aren't sure if this person has ever seen this thing and you want to know if they have antibodies against it to check if they've ever seen it, you need to have an antigen to check that. We learned this with our indirect and our direct ELISAs. Turns out indirect and direct are terms we're going to apply to all immunological tests, not just ELISA. All right. So be aware of that. We can use not just serum nowadays. We can use urine, CSF, tissue even. We can make suspensions of tissue and like break open the cells and whatever, and even saliva for some of this. So this is the basis for serological testing. Honestly, guys, this is like the basic breakdown of what ELISA did. So we already know these principles, but this is taking patient serum where they have all of these different antibodies from the serum, mixing it with our known antigen, and then mixing them together to see if there's a reaction. So in this case, we're looking for agglutination or precipitation. In this case, we don't see over here in our negative sample, we don't see any precipitation or agglutination. That's what's going to happen when our antibodies are sticking onto antigens, right? So that's all this is showing. The other side of it, we have our culture that we made. What is this thing that's growing in our culture? We don't know what it is. So we use antibody that we do know. So known antibody mixed with your culture sample, and then same thing, look to see if we had a reaction. This is the same concept of what drove ELISA. We're just not using a secondary antibody to look at it. We're just looking for agglutination or precipitation. If you guys have seen or done any blood type testing, this is exactly how this will work. You'll do this in like little wells and see if there's agglutination or precipitation from antibodies that C, A, or B, basically, or RH. So here are our methods that we will be talking about for our immunological diagnostic techniques. We already kind of mentioned agglutination and precipitation. We'll talk about the difference in a moment. Immunochromatography is a fancy way of saying antibody antigen with color change involved. Guess what? That's what COVID home tests are. So that's immunochromatography. It's just a fancy word for that. They call them laminar flow systems because they're flowing across something, fluid moving, and then a color change happens. These are just crazy fancy words for stuff that we are used to seeing. Then we'll talk about Western blot and fluorescent antibodies, ELISA, and in vivo testing. So antigen antibody interactions, we can see agglutination versus precipitation to start with. Agglutination is talking about whole cells. So you will agglutinate cells. Great. Precipitate will happen when we have soluble molecules. Either way, it's going to look like clumping in the bottom of the well. You're going to look for that versus like it stays suspended. So you have nice diffuse mixed suspension versus clumping. That's what both of these are going to do. Really interesting stuff, I guess. But like we do this a lot with blood typing. If you have agglutination, we are going to be causing that binding and sticking together of our cells versus our non-agglutinated mixtures. So moving on to our immunochromatography. This is our lateral flow tests. Again, the picture, I feel like describes it better. Now we know this is something familiar. Putting a sample into a little area and waiting for it to flow. Laminar just means like a solid surface. If you've ever seen, so there's these like videos you can see where like they'll like have this big old bag of fluid or a big old thing of fluid and they'll cut it open. And like the fluid comes out at first, you can tell that it's fluid flowing. But then once it's been going for a while, it just looks like it's sitting there stationary, like a bridge of fluid flowing out. They call that laminar flow. So it's like this, like really, I would say routine. I don't really know what else to call it, but it's not changing at all. It's just like all flowing, all in the same rate, all across that one direction in the same concentration. That's the idea with laminar flow. So we have that very controlled amount of flow of fluid across the whole entire strip of paper or whatever it is to get that going across your bands of antibody or whatever it is that's going to cause the color change when we interact with it. So what's actually happening at these bands is we have antibody antigen interaction. That's it. And a color change as a result. So it's immuno because of that antibody antigen interaction. It's chroma because of the color. And then the ography is just testing, okay? So that's all that's going on here. Don't read too much into it. Antibody antigen color changes. We've mentioned previously as well about antibody titer. This is just measuring the concentration of antibodies. We can use this to diagnose certain autoimmune disorders, as well as past exposures to certain pathogens. We've talked about that with HIV. So with serotyping, so this is all serology, doing antibody antigen interactions for diagnosis, but serotyping is determining the type of bacterial strain you have based on if it interacts with certain antibodies. For like if we look at, let's say, salmonella, there might be some species of salmonella, or we'll say serotypes because we're talking about serotypes, okay? There might be some serotypes of Salmonella enteritidis, which is a species of salmonella. Some Salmonella enteritidis have flagella, and some Salmonella enteritidis do not have flagella. It's just whether they have that gene or not, but they're still Salmonella enteritidis. So we can use serotyping to look for if they have flagella or not. Use antibody antigen interactions for flagella. And that's going to determine that this is Salmonella enteritidis serotype, blah, blah, blah. based on if it interacts. So that's all this is. It's like taking species of bacteria and getting more information about them. And now they get a new little name because of it. So that's essentially what a serotype is. Getting more information based on immunological interactions. Okay, so next is Western blot. Western blot is essentially using our same principle of gel electrophoresis, where we have our DNA molecules that are negatively charged. They're going to move to the positive end. The big ones are going to get stuck. The small ones move faster. Great. So we've got that in our minds. Now imagine that doing with protein. It's going to do the same sort of thing. Okay. So we don't have to worry about like the charge difference here, but imagine that they are moving to one end of the gel, and the bigger ones get stuck and the smaller ones move faster. Same thing. Same thing. So gel electrophoresis with proteins. And after you have spread out all your little proteins in those nice little pretty little bands that we get, same way as we do with DNA, we're going to use another electrical method to push it out of the gel onto a filter paper. That filter paper now has those little proteins stuck to it, and we can put antibody onto that and see if it binds to those little bands. We can't do it to the gel itself because it's a whole thick old gel. So we just put it onto that paper because it's nice and consolidated. And we can block the whole background with just something like milk or something, so that we're only looking at the bands and interacting with those with our antibodies. This is extremely sensitive, and it is extremely expensive because it has to be done by hand. There's no machines that do this for us. And but it's like if you test on your ELISA as having an interaction with HIV antibody, we will confirm with Western blot if we are at all unsure about that reaction. So if it seems like there may be positive, it's sort of weak, then we might confirm with ELISA, or sorry, excuse me, with Western blot, those results that we saw on ELISA, so that we can ensure that we're telling the patient that they're positive when they're actually positive, and not accidentally telling somebody that they have HIV when they don't, because they have some sort of weird ELISA cross reaction. So this will be so specific and tell you about the specific bands. So essentially, you have your gel that we're using on your electrophoresis, and then you have your little bands. So here's where your protein would be on your membrane. Again, this turning it yellower means we're putting milk on it so that we don't accidentally blot anything onto blank area of the membrane. These antibodies are going to bind to that protein area, which we couldn't see previously. That's why it's the dotted version. So now we can have antibody binding. And then, of course, go figure, we're going to have our secondary antibody so we can actually see that the antibody stuck there. So you have rabbit that you bred up to have antibodies against HIV. You put that onto your little paper. It binds to your patient's band. And now you put your secondary anti-rabbit antibody on there. And it's fluorescent or it's radioactively labeled or something like that. And then you can take a picture of it and see that, yep, there's a band here. And that's how that whole process will work. I'm not going to ask you about the steps. I just want you to understand how this works. Like, it's going to recognize that this patient has this specific protein based on the gel electrophoresis bands. And it uses antibody antigen interaction. So y'all might be up here sitting here thinking that I don't have my little cup. I'm not stupid this time though. I came prepared. I don't have my cup with the things. I'm not going to call out names. All right. I am, however, going to be calling out teams. So for our first, let me see if I can do this. Split view. So we can see our questions. All right, great. Are we having fun yet? Yeah. All right, so let's do this. So this is number 6. So this is Foxtrot. Anybody from Foxtrot can pick a question up here. Honestly, how about everybody from Foxtrot stand up? Come on, guys, Foxtrot. All right, guys, it's all you guys, okay? Any of you guys can pick one question up here and answer it for me. Or I'll pick it for you. That's pretty much what we're going to go with this. All right, I'll give you 5 seconds. 5, 4, I'm being generous here. With number 1. Okay, so the question is, everybody, what type of media is used to encourage the growth of a specific pathogen? And what is that called, Foxtrot? Want a specific pathogen to grow? You might use blah, blah media. What is it called? Exactly. So it's selective. So Foxtrot gets a point for that. Good job. So you guys can sit down if you'd like. Hopefully you guys don't get called again twice in a row. So Foxtrot got number 1. All righty. Next group. This is Charlie. So Charlie, stand up for me, please. Come on, guys. I know I can't, I can't just people listen. Yes. All right. Fox. Okay. Sorry, Charlie. Yeah, it's okay, guys. Charlie, what question would you guys like to do? Remember, I'm looking for general answers here. You guys know how this goes. Anything you prefer? All right, so let's go with number 2. And again, I'm looking for like general approach, okay? How are biochemical tests used to identify bacteria? So you can either explain like a general concept of how they work, or tell me of our 3 groups that we've already talked about. We've already talked about, gotten detail on 2 of those groups, which group they fall into. So I'll give you a hint on that. We had genotypic, immunological, and phenotypic. Which one would biochemical tests fall into? Phenotypic. All right, so that's going to be phenotypic. So we did this one. So these are good. So yes, that's going to be phenotypic, right? So phenotype, looking at observable changes in these bacteria, what they're expressing. Remember, genotype is always going to be looking at their exact genetic makeup. Immunological is going to be looking at antibody-antigen interactions, always. So if it doesn't involve antibody-antigen, and if it doesn't involve exact genetic makeup, it's phenotype. Okay? All right. Good job, guys. All right. Next, we have it's Foxtrot again. All right, Foxtrot, it's you guys again. Is there another question you guys would like to look at? 3, 4, or 5? Okay, so 5. An example of a commonly used immunochromatography test. COVID, exactly. So that's an easy one. That's going to be COVID, pregnancy tests, anything like that where you have that line color change. That's an easy one. So these aren't as hard as you guys try to make them out to be. All righty. This is Delta. Yes, Delta. Okay. And so you guys get a lucky boost because Hannah in her online group also is Delta in her group. So she's here to help you guys out if you need it. Any question that you guys would prefer? Number 4. Okay. So what is serology?
Use serums to identify antibody.
Yes. So that's good. Yes. So antibody antigen interactions. That is great. So you guys get a point for that one. Good job. So we have 1 question left. Who's going to be the unlucky phage type? Oh no. Okay. I'm going to give you a break, guys. We'll go with Bravo. Bravo group. Anybody in Bravo group? How is phage typing used to identify unknown pathogens?
Kind of like the Kirby Bauer uses the disc and.
It is a lot like that. So I'm going to give you points for this because Kirby Bauer uses the discs of antibiotic, whereas phage typing, we're introducing little bits of the viruses that infect the bacteria, but it has the same outcome with the clearing and everything. So yes, so I'm absolutely going to give you points for that because you get the theory of it. That's what's most important. Okay, so good job. Actually going to now I did all of that. Now I have to erase it for the next class. So good. So Foxtrot got 2. Charlie, Delta, Bravo. I should like write this on the back of my hand or something so I don't forget. Only Alpha didn't get called, right?
Right.
Okay. Bravo. And Echo, yeah. Okay. So yeah, Echo's like sitting there like, no, if you want to give us a point, that's, yeah. All righty. So back to this. Moving on with more of our immunological testing. This is going to be immunofluorescence. So now we're putting antibody that has a fluorescent tag, but instead of like what we learned about with ELISA and the color change there or whatever, we're literally imaging it usually with a microscope this time. So we can take tissue samples and get thin slices of them and incubate them with antibody. If the antibody sticks, then have a secondary antibody that recognizes that antibody is there, of course. Instead of an enzyme being linked to it, we have fluorescence. So it'll glow green or yellow or red or whatever. And we can take actual pictures of things. So this, when we look at this picture here, these are actually, these are cells. This is a bacterial cell. This is, it turns out syphilis. Okay, that's one bacterial syphilis cell. And it's coated in antibody that is fluorescent. So now we can take a picture of those cells because they've been made to be fluorescent. We use anti-syphilis antibodies. They stick on there, they're fluorescent. Now we can take a picture and show you, looks like your smear or your tissue sample or whatever it is has syphilis in it. Congratulations. You might need some penicillin. So these work the exact same way as our ELISAs do in the concept of it will be direct or it will be indirect. So we can use labeling based on that. So direct testing with fluorescence, does the patient have the antigen? Is the patient sick with the thing? Very direct question. Whereas indirect, does the patient have the antibody? And it's going to use basically the constant region of certain antibodies if they're present in the serum and use that for fluorescence. You guys don't need to necessarily know that. Just know that direct, we're looking for the antigen, and indirect, we're looking for the antibody, right? Okay. We know ELISA. I probably don't need to do too much with this one. Definitely know what each letter stands for, ELISA. I'm not necessarily just going to ask you about only the E anymore, right? We're adults now and we know ELISA. Enzyme linked. Again, you will never have to write it. So there you go. But enzyme linked immunosorbent assay. We have indirect versus direct. Again, indirect, we want to know if they have the antibodies. Direct, do they have the antigen? Do they have the thing? So that's all we're looking for. And color changes are going to be our positive results. You guys have seen these before, just depicting how indirect is going to work. You coat it with antigen first, then you add the patient sera. If they have the antibody or not, determines what happens next. If they are positive, they'll stick. Then we'll look to see if that antibody stuck. So we'll add our secondary. Then we'll add our substrate. Then we'll see a color change or not. That's all that's happening. We know this. This is not new. Direct is our sandwich method. Does the patient have the antigen? So essentially, you'll start with your antibody. You'll have your patient sample. So that's going to be positive. So then we can have our antibody, again, that recognizes that same antigen, but it also has our enzyme. And then we add the substrate and the color comes. And that's how that one works. Just less wells for this one for some reason. They didn't want to explain it as much, I guess. And then, you know, you get your samples and you can look at which wells are positive or negative and how positive or negative, right? Based on how strong that color is. And that's going to be based on your patient's antibody response if you're looking at antibodies, right? That's ELISA. Next, we have the complement fixation test. It's still antibody antigen interaction, but instead of using color change or instead of using fluorescence, now we're going to look at if complement is doing its stuff. So remember, complement is going to poke holes in things and cause our cells to release their contents. And so if you are having complement interaction, you will break open cells. And so what we do is we mix your patient sample and your maybe antibodies, or we'll have antigen mixed with your patient sample, depending on which one you're looking for. Whatever it is, we're going to mix your patient sample and the known antibody or antigen, and then mix it with some red blood cells. and then see if those red blood cells get lysed or not. If they lyse open, complement was active. That person has the thing or had the thing. So that's what we're looking for here. That's all we're looking for is lysis. So yeah. And then blocking of it as well. So anyways, in vivo testing, this is looking at real life response in live tissue to something. So this best test that we have, we already learned about it in the last chapter, was that skin test, the TB skin test. So putting tuberculin protein under the skin and seeing if you have your delayed response in the 24 or 48 hours later. That's all. So is that still immunological? Yes, because we're still talking about the immune reaction. We're still talking about antigen. We're not necessarily talking about antibody. We're talking about T cell response here, but it's still your immune system. So it still falls in this category. Oh gosh, don't you guys love specificity and sensitivity at this point? So it keeps coming up. You can't get away from it. Sensitivity and specificity coming back up again because we're doing our diagnostic tests, okay? Specificity versus sensitivity. Like I said, I always like to start with sensitivity. It's the easier one for me to understand because I feel like if you're really sensitive, you'll pick up on anything. So sensitive means you're going to pick up all of your positives. You're going to see every single positive, no matter how weak it is. You'll try to pick up as many as you possibly can. That's what sensitivity is. So you'll see all of the positives. There shouldn't be anything you're calling negative because you picked up all the positives. So we're eliminating false negatives. So you will not be calling something negative what is actually positive. So low false negatives for the sensitivities because you're picking up as many positives as possible. For the specificity, okay, this one, we are only binding to positives, okay? We will not be so sensitive that we'll accidentally be calling negatives positive. We're not having that. The only things that we're calling positives are positives. So this is filtering a little bit more, being more specific about the things that we are calling positive. So we are going to have a low false positive rate for these guys. So this is looking at these sensitivity. Okay, the first one we talked about, sensitivity, low false negative, because we want to pick up as many positives as we can. Specificity, low false positive, because we're not going to call things positive when they're not accidentally. So obviously you want both of these. You want a low false negative and a low false positive. You want everything to be as true as it can be. So you want both. They're not mutually exclusive. All right, now we're moving on to the genotype stuff. I told you guys this is going to be fast. The genotype stuff is all looking at literal genetics of the organism. Do they have the nucleic acid sequence? That's all we're asking here. PCR, we know that's going to duplicate DNA strands every round. So if you had one strand, you're going to have two. Everywhere that you have one strand, the next round you'll have two from that single strand. So we know there's going to be doubling every round. Amplifying the DNA. We can do this with all sorts of pathogens. With PCR tests, there's different versions of them. Real-time, reverse transcriptase, multiplex, where we have more than one thing we can test for in one setting. Pan-bacterial, we can do PCR just to say, like, is this gram-negative, for example? Or RT-LAMP, I don't even know what that is, okay? So that's on there, but I don't know what it is. Real time, you're getting fluorescent markers associated with A, T, C, or G. And as we use A, it flashes yellow. As we use T, it flashes green, and et cetera, and et cetera. So you can track whether you're using A, T, C, or G as we go. And a measure of that, if you're increasing in your usage of those colors, and you can track them the whole way through, you can know very early on in PCR whether you're actually amplifying or not amplifying. So you know in real time what your results are going to be. With reverse transcriptase PCR, we can take RNA samples and then amplify those. This is useful for RNA-based viruses. They don't have DNA genomes. We can convert it to DNA, which is more stable, and then use the DNA instead. Multiplex, like I said, this is going to be doing different organisms all at the same time. So you can use one PCR test to test for flu and RSV and COVID all in one sitting. Those are really complicated ones though. Okay, so next from PCR, we have hybridization. Again, we have probes that recognize whether you have this gene or not, right? So does this probe sequence match this patient sample? If it binds, that means they have the gene, that means they have E. coli. All right, so that's the idea with these. One example of this is FISH. This is fluorescent in situ hybridization. So in situ means we're using whole intact cells here. So it's not in vivo where we have in living tissue. This is in situ where we might have tissues that you got from biopsy or something like that. We don't need to break the cells open and get their DNA out. We can use the intact cells. So this makes it faster. And we'll look at this because it will turn fluorescent. So we can find cells that match the fluorescence essentially, but this is not based on antibodies. This is based on our DNA interactions. So the probe, again, with the same sequence for that gene, if it binds in that cell will be fluorescent. So that's what we're looking for. All right, so this is an example of FISH. This is peptide nucleic acid FISH testing for, you don't need to know this exact one, but essentially all we have is our blood culture. And then we have our, do a gram stain of that blood culture. We see staphylococci, but is this Staph aureus or is it something else staph wise? So we'll take that culture with our fluorescent probe and bind it on there and do some rinsing and then look at it under the microscope. And that's what they see. So it looks like this person's blood culture has fluorescent cells that bound with our probe that is looking specifically for genes associated with staph. So they have Staph aureus specifically. Cool. Whole genome sequencing. We can also now how quickly we can sequence entire genomes for our pathogens. It's so crazy how fast it is nowadays. So we can just shotgun sequence whole pathogens and say, oh, this pathogen, it's not only is it E. coli, but it has these resistance genes and it makes these toxins and whatever. So you can get a whole profile of your organism by sequencing its whole genome. So you get rapid analysis. If you have an outbreak of something and you have no idea even what it could be or where it's coming from, you might just do a full genome sequence, just to get as much information as you can all at once about that organism. Microarrays. Remember those probes that we're talking about with FISH? We have those little sequences that tell you that this organism has this gene for Staph aureus. Well, imagine you take a whole bunch of those little probes, pop them onto this massive plate with like 365 little dots. Each dot has a different probe for a different organism. This dot for Staph aureus, this dot for Staph epidermidis, this for strep, this for E. coli, this for whatever. Each dot is something else. Now take your patient sample, mix it all over that, and see which dot it binds to. In fact, you can do this to the point where you would not necessarily look for each individual organism, but for different strains of these organisms. So you might have a whole plate just for E. coli. Is it this toxin, this toxin, this toxin, this toxin producing type of E. coli? And is it resistance to this for E. coli? And then determine all of that information from one test, one incubation with that plate. So that's using genetic sequences. Again, we have DNA probes that are going to match with DNA sequences in that patient sample. We usually use fluorescent labels and then a machine will analyze it. So look here, we have all these dots on here with all different kinds of samples, different versions of E. coli, or we're here talking about infection all the time. What about different kinds of cancer? So here we have different versions of breast cancer. If you guys don't know, there's like so many different versions of all of our different kinds of cancers that it's ridiculous. So whatever could be causing your breast cancer, is it the BRCA1 gene defect? Is it the MTHFR gene being this way? Is it this gene? Is it that gene? Is it this version of this gene? Is it the version of that gene? What mutation is it specifically? Make DNA probes for each mutation type, put that patient samples on there and see if their genetic sequences match what's on the plate. And where it does, when it does match, tells you information about that. That's microarray. So we can create basically a lab on a chip where we can look at not just the DNA microarray of certain diseases, but also immunological, potentially all of that on one chip and get immunological and genetic data from that one chip. This is more useful for rural settings and for less, more underdeveloped countries where they don't have labs. So they can do this testing in a smaller setting using all of these techniques, whether it's immunological or genetic, to get diagnoses for their patients to treat them better. So that's the idea, developing countries and rural areas. All right, so we are out of the realm of genetic now, and we're moving into others. Okay, these are other categories. This is mass spec. You've probably heard of mass spec in passing. It's not something people talk about a lot. A version of mass spec is MALDI-TOF. So don't ask me to tell you what it means. I used to know, but the TOF of the TOF means time of flight. And what that means is we're going to take your patient sample and bombard it with basically radiation, usually lasers, but it's going to cause your patient sample to ionize. And then the machine determines, this is chemistry, you guys, and physics and chemistry, right? That's not my strong suit, but you're going to bombard the molecules of your patient sample and then look at the ions that are coming off based on how long they spend in the atmosphere, their time of flight, and their charge and their mass. And that will tell you their exact molecular makeup. So you know their exact chemical composition, their molecular makeup, and know exactly what that molecule is based on breaking it apart and measuring the parts. That's what MALDI-TOF does. So it is very, very molecularly based, like sub-molecular molecule based almost. It's useful if you really need to know exact like chemical compositions of things. If somebody has ingested something, chemical, maybe you know, an attempt for committing suicide or something like that, and you don't know what drug they took, you can take a blood sample from them and subject it to MALDI-TOF, and it'll give you a chemical profile of what's in their bloodstream. So you might be able to delineate what drug it is that they've exposed themselves to. Of course, there's processing involved and all that, but that's the idea. You won't just look necessarily for chemicals. Of course, you can look for microbes as well, but that's where it's most commonly used, is for chemical analysis. We also have imaging. These are obvious, right? MRI, CT, PET scan, and X-ray sometimes, especially if we're talking about tuberculosis, we're looking for cloudiness in the chest. These are used for diagnostic techniques as well, for infection, as well as other things. We already know for cancer, these are obvious, but infection can show you sites where you might have abscesses and things like that. So we don't have to do exploratory surgery. Now we know where you might have the abscess, and we can do a needle biopsy or something for those things. So that is, then we can do other tests beyond that. So guess what? There's more questions. We're not going to do the group thing this time, okay? We'll just go through these ones because we have the time, then we can just go to lab. What type of test asks, does the patient have the antibody? If we're asking for antigen, that's direct. But if we're asking for antibody, that's indirect, right? What does ELISA stand for? That's enzyme linked immunosorbent assay, right? What test relies on the lysis of red blood cells if the sample has the antibody? That was the complement test. What diagnostic testing trait focuses on only certain targets, thus reducing false positives? If we are only focusing on certain specific targets and not accidentally calling something false positive, then what we're going to be reducing by the false positives, we're going to be calling that specificity of that test. That was a weird way of me wording it, but yes, that's specificity. What type of genotypic testing utilizes the fluorescently labeled nucleic acid probes on intact cells? That was FISH, right? So that's intact cells, fluorescent labeled nucleic acid probes, all right? So before I forget, we had 2 points for Foxtrot, 1 for Bravo, 1 for Charlie, and 1 for Delta, right? All right. Go to lab.
Question.
Yes.
Is optimization and coding the same thing? Or is it like different?
No. So opsonization is essentially creating like little molecular handles for phagocytosis to happen. So phagocytes, when they're able to grab onto those handles better, that's aiding that phagocytosis. That's opsonization.
just keep it from,
It's just like you're literally just coating it, like if you were like smothering your dog in a blanket and now it can't run around the house. That's basically the idea. Yeah.